Welcome back, guys. We're going to move a little forward here in our protein fractionation lecture. Again, we had started from cell lines, uh, characterized them by flow cytometry facts. We had broken them open with the different centrifugations and fractional separation. We talked about cell-free systems, and now we're bringing in to the protein level. And once you have a pure protein, it can be sequenced, you can make antibodies to it, you can use that protein to find protein partners. It's a very, this is a path that allows you to study, key proteins. Is this protein associated with a disease or not? If so, is it causing the disease, or is it downstream of the disease's cause? So you get a chance to dig deeper still. So all those fractions that you collected in your fractionation, uh, your centrifugation and different density gradients and things, now the payoff starts here. And again, remember, we can test the proteins at absorbance 280 or the Bradford. And eventually we're going to bring in electrophoresis. So just in the back of your mind, realize that this purification has to be confirmed. And one way is we actually visualize the proteins through protein electrophoresis, which is the next, after we're done with the fractionation, the next block of material there. So I just wanted to kind of put that in perspective. So I kind of left off with this flow diagram, and it's just reminding us that purity can be low, medium, or high. And so depending on what we can do, we may have to muddle through and do multiple tests to try to get higher levels of purity. And just by the way, ion exchange and affinity are the most common used chromatographic strategies for one-step purification. So this is really the key, is the one-step purification. And of course, it won't let me go back. But anyway, the affinity, like I said, if you've got an antibody to a protein, then most likely you can pull it out of solution. So that's the best case scenario. And you're saying, well, if it's a disease, how might you have an antibody? Well, if it's a virus or a bacteria, most likely their serum, if they're a sick individual, especially if they're survived it, they're going to have antibodies in their system, and those can be purified. And then sometimes you can purify a protein from antibodies that exist. If there are proteins that are closely related, sometimes antibodies will cross-react. So depending on the different needs of high, medium, and low purity, depends on where we'll go. So let's start with size exclusion chromatography, liquid chromatography. So right away, it's keying in on protein size and exclusion. That means things are prevented from going into an area, right? You're excluded from going into an area. There's a fence or electrical fence. You're excluded. So what happens is there's specialized beads that are very porous, and these little let me get my little red ink here. These little black kind of curlicue looking pieces of popcorn actually are beads that have pores in them. And you can order from the Walmarts beads that have different sizes drilled. Imagine little miniature bowling balls, but the finger holes go all the way through. So the big concept is this: if you're a small protein, and it's shown in blue here, they will travel and it could be through gravity fed. And remember, this is all liquid as well. So you've added the proteins in a reservoir up here in liquid, and they're coming into this. And these proteins are flowing across, kind of like water across rocks and rapids, except the small proteins will actually journey into the holes in the beads. And you can imagine if you stop at every bead and kind of flow through it, it's going to take a certain amount of time. Large molecules are too big for certain types of beads, and they flow directly around. And so large molecules will end up moving through versus smaller molecules that will move slower. And so again, depending on what you're doing, you can order the different size beads. And so now we see size exclusion. Large proteins are excluded from spending time in these beads. These beads must be inert. They cannot have any kind of protease activity, any way to chew up proteins. And so and again, we may not know what the size of the protein is, but we'll certainly have a fractionation that will separate large to small proteins if we do a fractionation and collect the drippage that's coming through this column here. And so this is another cartoon showing the beads that have pores in them, and here are 4 or 5 proteins. And again, the large proteins will flow through, the smaller ones will spend time. So it's showing the big blue giant proteins are flowing around. The other proteins are flowing into the beads. Okay, so the large, and they call this, I thought it was blue, they called it green. It flows faster. The red, yellow, medium size get trapped in the pores, and the very smallest stay trapped longer. So there's a retention of the smallest proteins, the middle protein, and the large proteins are coming out first. So there's an order at which proteins in a mixture will go through a size exclusion column. The size of these is usually 6 foot, and you're thinking, why would it be so long? Think about it. The longer you let the proteins flow through, the more likely they're going to bunch together. And all the large proteins will be kind of moving together. The medium protein moves together. If it's like 6 inches, you're not going to get a good separation. So you get a nice resolution the longer this column is. Okay, and again, you're fractionating here. There's liquid coming through, you're collecting with tubes, and you're then figuring out by Bradford or absorbance at 280 what fractions have what proteins. And here they are, this concept here. So here's the blue from the previous thing, and you see the black proteins are hanging up in here because they're the smallest, and here's the medium. So if we go here, the first, second, third, fourth, and this may be opposite because these would come first. So that may be a mistake because I think the large ones come first. So anyways, you're tracking, you get protein peaks, okay? And so that means that these can be excluded. You don't have proteins in there. Of course, this is perfect in a card. So now you throw away, and now we're dealing with 3 fractions, say 3 mils. Each fraction's a mil. We can do Bradford, or we can do the absorbance at 280. So the second, so once you have, say you know your protein is the larger protein. We got 3 tubes. And you're saying, well, there's a bunch of large proteins, right? It's not just one, even though the cartoon showed one. There's going to be a bunch in a cell, okay? Bunch, thousands. So can you separate the large proteins by their charge? And that is what ion exchange chromatography does for you. You have two types of resins. These are inert polystyrene, polyethylene resins, ion, and they have a positive charge. So the company actually imparts a positive charge to these resins. And so if they have a positive charge, what are they going to do? They're going to try to grab on to the proteins that have a lot of negatively charged R groups. So that's where the charges are coming from. So when you take biochem, you'll have to memorize all the R groups and who's positive and negative. Another type of resin is the cation exchange resin, and these have a negative charge, and they're going to bind positive molecules. So you can have two different columns and pour your samples through and separate them that way. And you can also have pores in there as well, so you get the charge and a size effect with ion exchange. So this is the positively charged resin, and here are the different proteins, and the matrix is positive. Only the positive proteins run through, because if they have a positive charge and the resin is positive, then guess what? They will be repelled. These will attract negative proteins. So in this cartoon, the black proteins, the small ones, have the positive charge, and so they go through first. The rest of the proteins are stuck attached to the resin. Okay. And so what happens in this, just a cartoon showing that we'll apply the resin, things are being caught. And so how do we knock off the one stuck to the resin? Well, we can change the pH or salt them out, and that will allow the proteins, the charge forces, to break apart. And so here's the solvent throw. Here's positively charged beads catching negatively charged proteins. The positive proteins flow through. And then here's the solvent flow with the beads. And that of course, we like to give multiple names. Gel filtration is also size exclusion. So we try to give multiple names to the same thing. Okay, so that gives you an idea. So again, just put in your notes, because I don't have it in a slide there, that you can alter the conditions and break the charge and release the other proteins. But in that cartoon, the black proteins were already pure for you. So, but again, it's not going to happen that way. Hey, fussy dogs. So the final main liquid chromatography is called affinity chromatography, and it's a separation based on some type of conformation. Resins can have ligands attached on their surface that can be very specific for certain compounds. These ligands could be antibodies, they could be metal, they could be other kinds of chemical materials that bind to a protein here. So here's a protein that binds to the little Pac-Man bind, and then you can, here's a competing protein that you add to the column and knock these off, and now these are pure. And so this is called affinity because the resin has a great affinity for the chromatography. And that is especially good with antibodies. So you can actually buy a resin with an antibody, or if you have an antibody, you can buy a resin you can do a chemical reaction and attach it to. So here it is. Here's that. You can compete with this ligand, knock it off, and so free ligand can bind it as well, or you can introduce another protein. Two ways to knock off an affinity resin, and what they're not showing is all the other 9,999 proteins have already been washed through the column, and so they're gone. Okay, those proteins are gone, and what's left on the column is the very specific bound little pink Pac-Man that you then elute by adding the ligand, and that competes and knocks them off, or a competing protein that takes the place, and it must have a higher affinity, right, than the Pac-Man, or this won't work. And so now we got pure Pac-Man in any case. And this just shows affinity chromatography. And here's another one. You bind, you have an affinity wash. Look, all the other proteins are dropping out. Then you do an elution, and then you have some type of salt or other mechanism that you can knock the red, looks like the red proteins are bound, and you can knock those off at that point. Desalting, and you can change pH or salt, and you can get rid of those. So you can, like I said, purify proteins based on their sequence, based on if they bind a metal, based on if they have an antibody, various characteristics. Hydrophobic proteins can be bound with hydrophobic resin. Hydrophilic proteins can be bound with hydrophilic resin, and we'll talk more about that. So this is a cartoon showing antibody affinity columns, and of course we loaded near pH 7, we maintain the structure. Anytime you get too acidic or basic, you can actually change protein's conformation or shape. And so here's the protein in red that's recognized, and here's the protein recognized by this antibody. And so you have shh, quit talking. Quit talking. And so you can wash through and then change the pH and drop the proteins off. And so there it is, fraction 1, 2, and 3. And then hopefully we have pure proteins at that point. So this is the different kinds of affinity. You can use an enzyme that will bind substrates. You can use antibodies to bind an antigen. You can actually purify virus from a patient's fluid. You can purify cells, kind of like the FACS, where you targeted proteins on a cell. You can actually immunoaffinity purify cells if you have an antibody. Lectin will bind to oligosaccharides. Nucleic acids will bind to proteins such as histones and polymerases and binding proteins that bind to DNA. And wow, I don't know what that was. But anyway, you have a choice in metal and some other methods to bind and use in the affinity column here. And this is just another cartoon showing the fractionation. And so here in the ion exchange, we let the proteins come through, and then we salt them out, and we found if it's an enzyme, we found that in these fractions here. Okay, and then here's size exclusion, and we purified this fraction as well. And then here's the affinity. So we get all the other proteins and our one protein. So in this cartoon, here's the one protein. And here are all the other proteins. Each band represents a different protein. And here's some weakly binding proteins, including our target. And then 3 is the strongest binding proteins on the affinity column. And now we theoretically should have a pure protein that we manipulate further. So I'm going to end this with part 2 here, and we'll pick it up with high pressure / performance liquid chromatography. So this is a very expensive machine, $100,000 machine, but it's the ultra pure mechanism for further purifying proteins from fractions here. So come back and we'll talk about part 3 here coming very soon to you.